Welcome to this tutorial on SGD's JBrowse genome browser. In this video, you will learn about JBrowse data tracks and how to add and remove them. JBrowse represents annotations and experimental data in the form of data tracks. The 7 tracks currently shown here are enabled by SGD by default and represent information such as RNA polymerase occupancy, genes whose expression is affected by glycerol, and the chromosomal features and ORFs of the S288C strain. In addition to these 7, SGD's JBrowse has other data tracks that show things like nucleosome occupancy, polyadenylation sites, transcription factor binding sites, and a whole lot more. To add more data tracks, use the Select Tracks button over here. This will open up a list of all the tracks that are currently available in SGD's JBrowse. The side menu, over here, partitions these tracks into certain categories, such as assay type, general biological topic, and more. Let's say that we're interested in where nucleosomes are positioned. To find data tracks that concern nucleosome occupancy, enter Category and click on Chromatin Organization. This returns 40 data tracks that have experiments or data related to chromatin organization. To sort through the tracks in this list, click on any of the column titles here to sort column values alphabetically, or filter the list by using keywords in the search box here. Searching for nucleosome, for example, returns 13 tracks from the list of 40 that contain the word nucleosome. To add data tracks to the main display, simply check the box next to the data tracks of your choosing, and then click on Back to Browser. The data tracks you selected will be added at the bottom, here. To move them around somewhere else on the main display, click and hold on the track title bar and drag it to the position you'd like. To remove data tracks, simply click on the X button, or open up the Select Tracks menu, clear your filters with the Clear All Filters button, and open the currently active section of My Tracks. Then, uncheck the boxes of the tracks you wish to remove. I'll remove all the tracks except for the reference sequence track, the S288C chromosomal features track, and the nucleosome occupancy tracks we added earlier. To find more information about the tracks you have on display, open the Configuration menu that's located in the track title bar and click on About this track. This will open a window that gives us a little background about the track, such as the source, authors, and institution involved, the kind of experiment or assay performed, and where the original dataset can be downloaded. Here, we see that this track is about intrinsic nucleosome occupancy in vitro, while this track is about in vivo nucleosome occupancy in the presence of galactose. In the promoter regions of the galactose metabolism genes Gal1 and Gal10, it appears there is mild nucleosome occupancy under normal conditions, but very low nucleosome occupancy in the presence of galactose. Thus, these data tracks suggest that the cell wants to express these genes when galactose is present. This concludes our tutorial. If you have any further questions or comments, please contact us at sgd-helpdesk@lists.stanford.edu.